A worm fired a harpoon longer than its own body. Not a metaphor, not a defense mechanism. A precision strike measured in a London laboratory, clocked out under 1 second. Ray Gibson documented it in 1972. A ribbon worm, Linnaeus ruber, 4 centimeters long, resting motionless in a specimen dish. Nothing about it signals danger. Then eversion. A pale, sticky tube launches outward from the worm's anterior end, past its own body length. Contacts prey, adheres. Gibson recorded it plainly. The proboscis can be evaded with remarkable speed, sometimes in less than 1 second, and may exceed the body length by several times. Several times. That is not a rounding error. The mechanism is hydraulic. Inside the worm's body sits a fluid-filled cavity called the ventricle. When the worm contracts surrounding muscles, internal pressure spikes. The proboscis, folded inward like a glove finger turned inside out, erupts outward. No skeleton, no rigid structure. Pure pressure released in a fraction of a second. A gloved hand holding a ruler beside a specimen tray tells the story numerically. 4 centimeter worm, 6 centimeter strike. The prey does not move again. In Nemertean species, the proboscis carries a stylet, a hardened, needle-like structure. Alexei Chernyshev at Russia's Zhemansky Center of Marine Biology described it in 2015 as functioning as a hypodermic needle, injecting toxins that can immobilize prey within seconds. A soft-bodied animal with a venomous harpoon. Retractable, reusable. These worms are not rare. They live in intertidal rock pools, in barnacle-crusted stone and shallow salt water, where seaweed smell hangs at low tide, in numbers large enough that you have almost certainly stepped over them. John Norenberg at the Smithsonian published a comprehensive phylogenetic analysis in 1993, establishing that Nemerteans have carried this mechanism for hundreds of millions of years. The weapon predates almost everything that has ever hunted on this planet. But the proboscis is only the beginning of what makes this animal strange. There is a number, a specific, documented fraction of a body, below which the biology of this worm should stop working entirely. It does not. That number, confirmed in controlled laboratory experiments, is coming. The easy answer is hydraulic pressure. Squeeze a tube, fluid moves. Simple. Sufficient. Most biology textbooks treat the ribbon worm proboscis exactly that way. A muscular tube under pressure, built like a water pistol. Case closed. Except Gibson documented something in 1972 that a water pistol cannot do. The proboscis does not push outward. It inverts. The tissue turns itself inside out as it extends, the inner wall becoming the outer surface, folding through itself at speed. That is not hydraulics. That is a structural transformation happening faster than the human eye can track. A squeeze bottle cannot invert its own lining. Not in under 1 second. Not repeatedly without tearing. The muscular architecture required for that motion is entirely distinct from simple pressure ejection. The worm maintains 2 separate systems, a retractor muscle running the length of the proboscis, and a ventricle, a fluid-filled cavity that generates the driving pressure. Those 2 systems work in opposition, simultaneously. One fires the weapon, the other controls the depth. No textbook diagram captures both at once. Then there is the regeneration claim, equally dismissive in most sources. Worms regrow, common in invertebrates. The highlighter goes under that phrase and the page turns. But Strand and Sundberg, writing in Zoologica Scripta in 2007, measured something that does not fit that sentence. Fragments of Linnaeus sanguineus, representing less than 1/200th of the original body volume, regenerated into complete individuals. Brains, gut, proboscis, everything. 1/200th. That is not a worm regrowing a tail. That number sits at the edge of what cell biology currently explains. The textbook phrase covers earthworms. It does not cover this. And if the proboscis itself can be rebuilt from a fragment that size, rebuilt with full mechanical function, then the weapon is not just fast. It is almost impossible to permanently destroy. Which raises the question the next chapter has to answer. What exactly is driving that reconstruction? The mechanism is real. The hydraulic architecture works. Gibson documented the Rinchicole in 1972, a sealed, fluid-filled cavity running the full length of the body, and the physics hold up perfectly. No muscles required. Compress the cavity wall, pressure spikes. The proboscis inverts outward like a glove finger pulled inside out. Elegant, complete, case closed. Except it isn't. Turbeville's later ultrastructural work confirmed what Gibson had noted in the margins. There is no muscular homologue to this system anywhere in vertebrate biology. Not a simplified version. Not a distant analog. Nothing. The Rinchicole is architecturally alone. That should be the strangest thing about ribbon worms. It isn't. Here is where the chapter shifts. In 2007, Strand and Sundberg published a paper on Linnaeus sanguineus. The same species. Buried in the methodology was a fragmentation protocol. Controlled cuts, measured fragments, and a result that required a second reading. Fragments representing less than 1/200th of the original body volume regenerated into complete individuals. Not partial recovery, not scar tissue, complete individuals. New gut, new anterior-posterior axis, functional nervous system. Strand and Sundberg wrote it plainly. Some Nemerteans can regenerate from fragments representing less than 1/200 of the original body volume. Consider what that means structurally. Planarian flatworms regenerate too, but researchers have mapped the mechanism. A population of pluripotent stem cells called neoblasts, identifiable under electron microscopy, distributed through the tissue. There is a blueprint, and science has located it. In Linnaeus sanguineus, no equivalent population has been characterized. Eduardo Zatara's 2019 cellular analysis of Ramphigordius documented stem cell mobilization patterns during regeneration. Mobilization from somewhere. The cells move. They organize. An anterior-posterior axis forms at the correct pole every time. But the source population remains uncharacterized. The organizing signal remains unmapped. The question isn't how the proboscis fires. That answer exists. The question is this: a fragment smaller than an eyelash reforms a brain, a gut, and a complete body, and science cannot yet locate where the blueprint lies. Here is the number: 1/200th. Not 1/2, not 1/10, 1/200th of a ribbon worm's body contains enough biological instruction to rebuild the entire animal. A fragment smaller than an eyelash, invisible without magnification, regrows the brain, the gut, the sensory organs, and the proboscis. Strand and Sundberg confirmed this in 2007, working with Linnaeus sanguineus in controlled fragmentation experiments. They cut animals into precise fractions, logged the outcomes, and found a threshold. At 1/200 of original body volume, regrowth was reliable and complete. At 1/201st, nothing. The tissue sat unchanged. The line was sharp. That sharpness is what stops researchers. A gradient would be manageable. Biology softens at edges, but this boundary does not soften. It holds documented, repeatable, and unexplained. Eduardo Zatara's 2019 cellular analysis tracked stem cell mobilization during regeneration in Rhamphogordius sanguineus. The stem cells moved. They organized. But no one has mapped the signal that triggers the organizational pattern itself. The instruction that tells a fragment which end becomes anterior, which cells become cerebral ganglia, which tissue becomes the sealed hydraulic cavity that powers the weapon. The brain regrows first, 2 faint lobes flanking the midline. Then the gut, then the ventricle, the fluid-filled chamber Gibson measured in 1972. The proboscis assembles last, and when it fires for the first time, it fires at full length. Under 1 second. The same strike from Chapter 1, rebuilt from tissue that had no weapon in it. The loop closes. The strike was real. The mechanism was hydraulic. The regeneration was documented. The threshold is confirmed. The explanation for why 1/200th works and 1/201st has not been found in the tissue, in the genome, or in any mapped stem cell population as of the most recent published literature. The animal lives in tide pools on every inhabited coastline on Earth. It looks like a thread. By every mechanical standard, it is something science has measured but not solved. Some records get filed before the explanation arrives.